Alright guys, in this video we're going to go over chapter 11 and the beginning two sections on microbial genetics. And so it starts out with talking about the blueprints of DNA, that bacteria have DNA and prokaryotes in general have DNA. Eukaryotes are going to have DNA, so what is true for what happens to the DNA in say, E. coli is similar to what would still happen in a eukaryotic cell in regards to DNA, the DNA structure itself. So the blueprints are the same. So let's talk about the functions of genetic material, but first we got a case study, and it is Mark. And Mark is 60 years old. He's also got type 2 diabetes, so like our previous person before. So he's got it under control, diet, exercise. He was doing some gardening because it's springtime, it's time to do gardening. And he scraped his leg while walking through some blackberries. And he kept working all day, and he didn't clean the wound and treat it with antibiotic until later that evening. So he waited until late at night to finally clean his wound. So over the next 2 days, the leg became red and swollen and warm to the touch. So he's definitely got some kind of infection. Sore on the surface, but also deep in the muscle. That's concerning. After 24 hours, Mark developed a fever and stiffness in the leg. That's some serious stuff. We need to do something about that. So he felt increasingly weak. He called a neighbor, and now he's in the ER. Good. I'm glad he's finally getting some treatment. Let's figure out what's going on with Mark. Okay, so the basics. We mentioned this before in the previous chapter, but let's go through central dogma. It sets off talking about DNA, and that DNA has two functions. One is for holding all of the genes that an organism needs and that needs to be passed down to its offspring. So this genetic blueprint that gets passed down to offspring. And then the second job of DNA is to regulate proteins. So DNA is going to be like we talked about in the previous chapter. We need DNA to make the RNA, and then RNA to make the proteins. So these three steps, DNA to RNA to proteins, is called central dogma. And then there are the steps in between when DNA writes the message. That's called transcription, and we'll go through that. And then we get RNA, we get the messenger RNA. And then messenger RNA is going to go out in the cytoplasm. If it's not already in the cytoplasm for a prokaryote, it's going to go out in the cytoplasm for a eukaryote. It's going to meet up with the ribosomes. And from there, it gets translated into, eventually, using transfer RNA and the ribosomes, into a protein made out of amino acids. So these three things, DNA to RNA to proteins, is called central dogma. And the steps to get from DNA to RNA is transcription. The steps to go from RNA to proteins is translation. Just because we have all these genes, again, does not mean that we're going to express them. We conserve genes, and we only turn on genes when we need to. This is key. Otherwise, you'd be wasting all of this energy doing all of these things that you don't need to do. Only do what you need to do, conserve the energy. So depending on what's going on, it talked about how you may turn on that genotype to show possibly different phenotypes. And the phenotypes are what's actually getting expressed, what shows up. So depending on the environmental condition, and I can't remember the exact temperatures, it was like 20 something, maybe it was 28, and maybe this was 34, I think, maybe, degrees Celsius for these guys. 27 degrees, 28 and 37, I was close. So the cooler temperature for SMAR, it expresses the red pigment gene. Otherwise, if it's not at that temperature and it's at a warmer temperature, like in the human body, it doesn't express that gene at all, and it keeps its normal lack of pigmentation development for SMAR. But if it is in that specific temperature range, it will express that gene and produce red pigments. So you got genes for different things, and you don't always turn on that gene. Just like if you have the gene for breast cancer, for example, doesn't mean you're going to get breast cancer. It doesn't mean you're going to express that gene. You may or you may not express that gene, and different things can affect gene expression. So genotype, the genes you have, does not necessarily mean that gene is going to be expressed. But when the gene is expressed, that's your phenotype, what actually gets expressed and shows up. They also posed in the textbook some really good ethical questions that I think all of us should probably be thinking about now in the age of everyone can just submit your spit to a company and they can crank out your genetic material. What happens with that information? That's some serious information to give to a company. And so they were bringing up questions in the textbook. Who's in charge of that data? What if that data goes to your insurance company? Can they deny your coverage because you have the breast cancer gene, even though you're not showing signs of breast cancer? Will they exclude you? So they mentioned the Genetic Information of Non-Discriminatory Act that happened in 2008. And that has to do with health insurance and your job, your employer. Your employer can't discriminate against you because maybe you have the genes for cystic fibrosis, whatever. But it doesn't cover, GINA doesn't cover life insurance, disability, long-term care insurance. So there's a whole bunch of other things that you just got to think about. And it's a lot of information, something to think about. We're going to move on because we got another section that I want to cover in this video, and that is DNA replication. So with DNA replication, when it copies itself, and it will copy itself if you're a eukaryote in the S phase, when we had interphase and there was G1, there was S, there was G2, and then you entered into mitosis or meiosis. S phase is in eukaryotes when it copies itself. Otherwise, in prokaryotes, we have binary fission, and that's where it copied itself. So when it copies itself, it's called DNA replication. And out of DNA replication, there were three different theories. We weren't exactly sure how this happened. So there were three theories. One was conservative. And with the conservative one, we keep all the good stuff to ourselves and we create a brand new double-stranded DNA. So all of this is new. So we keep our original and we use our original as the base to make a entirely new double-stranded DNA strand. And then this new one, make a new here, and here's more new and new with conservative. So the most conservative you could possibly be. Then we have semi-conservative. So halfway, we're halfway. We half keep one and then we get a new one. So here is the new one. And then we keep one half of that double-stranded DNA is old. And so we mix old with the new and old with new. So it's split in half. So it's not quite as conservative as full conservative, where an entirely new strand is made up, a double-stranded DNA is made up all brand new. This one, we split it in half, and half is new and half is old. That's semi-conservative, and we're going to show why that's really what happens. The third theory of the day was called dispersive, and with dispersive, we take the entire genome and we bits and pieces of it is how we think that they were copied. It was copied in little chunks. And they weren't separated out. They were just kind of chunked in together throughout the double-stranded DNA in just random spots. So we have new, old, all on the same strand of DNA. This is dispersive. So these are the three theories: conservative, semi-conservative, and dispersive. So let's figure out which one that was. And there were two scientists, Messelson and Stahl, and they designed an experiment to prove which one out of the three it was. So we have the three theories up here: conservative, semi-conservative, and dispersive. And what they did was they used isotopes of nitrogen, because we said DNA is made out of nucleotides. Those nucleotides include a phosphate and a sugary backbone and a nitrogenous base. So nitrogen is in every single one of those bases in there. So you need nitrogen to make new DNA. So every time DNA replicates, it needs nitrogen. Where I'm going with this is they used isotopes. So back before you had this class, you had some sort of intro science class, whether that was Bio 1010, Majors 1107, or even chemistry Hopefully you covered isotopes, where we said every element has protons and it has electrons. Protons are positive, electrons are negative, and they balance each other out. And then there were neutrons that hang out in the middle, and those neutron numbers could vary. Those are called isotopes. So here we have some cells, E. coli, and they are in this one that's got 15. So it's an isotope, nitrogen, the 15 version of it for this nitrogen, which is not its normal version at 14. So we have this 15 heavy isotope of nitrogen. And so every time this E. coli is replicating in this pool of heavy nitrogen, they're going to replicate and they're going to pull in that nitrogen that it's surrounded by to build more DNA with it. And when they do, they end up at the bottom when they spin down the cells and they harvest the DNA and they spin that stuff down the centrifuge. This is where it ends up in the column right there. And here is our double-stranded DNA strand created with this heavy nitrogen, created with the 15. And then here it is, here and here and here for all of our three controls. So all of our three controls have this heavy nitrogen. And we're going to take those cells and now using those cells with the heavy nitrogen, we're going to put that in a different solution. So we're going to take those heavy nitrogen E. coli and we're going to put them in a different solution, one that has 14 now, so a lighter nitrogen. So now those heavy ones can pull in the lighter isotope of nitrogen and see what happens. So we're going to pull it in and we end up with 14 and the 15. So if you can see this, because if I doodle over top of it, it's going to cover it up. But there is one old and one new, and one old and one new on the DNA strand. So we got one old strand, one new strand. And the way this worked, when they weigh them out on the columns, we are going to have the old and the new are kind of going to be in between. They're in between. They're in between. If it was conservative, you would have both strands of the DNA. If it was conservative, it would have been old, old, and new, new. And that's not what happened. So we immediately say that it wasn't conservative. It can't be conservative based on the results we get from this experiment right here. Let's continue on because we got to figure out, is it semi-conservative or is it dispersive? How do we figure this out? So now we're going to take these cells and we're going to put them back in and do the same process over again. Let them replicate. And when they replicate, let's analyze the DNA inside of them. And so when they did, they replicated in this strand, old, new. This strand is old, new. And these two, remember the old and the new split apart this round because this is like the grandkid version of it. This would be like F2 version. And from F2 version, now we've got two new strands here. So we end up with a split. And so we've got the mixed strand is here and the two new strands are up here. That's what we ended up with. So we know this is correct and this is not correct because we ended up with two different strands. We ended up with two new ones here and we ended up with these semi-conservatives here and here. So it is semi-conservative. That is how DNA replicates. It's not conservative and it's not dispersive. And that is thanks to the work of Messelson and Stahl. Okay, so how do these guys replicate? We're going to go through the process of replication. They are going to need to use energy to do this. So we've got this GTP molecule. It's similar to ATP, that it's going to be an energy currency kind of molecule. Let's go on to the steps of replication because that's one of the most important pieces in this section. I want us to know it, love it, and feel at least comfortable with at least the three basic parts of it. So with DNA replication, the first step is initiation, then there is elongation, and there is termination. These are the three basic steps. So what do we say when we mean initiation? So DNA is all coiled up, right? Because we said it was DNA wrapped around histones and they had to be tightly packed into chromatin in these chromosomes. And if it's really tightly packed like that, how in the world are we going to get into it to uncoil it and copy it? So the first thing we have to do is use I'm never going to say this right. Topoisomerase. Yeah, there we go. Topoisomerase. this guy is going to help uncoil DNA and loosen it up, because we said it has to get loosened up and then we can find those grooves and these things can attach on. And now we can use this guy, helicase, and he's going to split the DNA. So our topoisomerase uncoiled, so it's this guy, he uncoils. Then helicase, so we've done this guy, we're headed to helicase. Helicase is this purple one, and he comes along and he's a wedge, and he wedges in the DNA and breaks those hydrogen bonds that we said keeps DNA together. And remember we said A's and T's and C's and G's. A's and T's had 2 hydrogen bonds and C's and G's had 3 hydrogen bonds. So it was easier to break at the AT site with the 2 hydrogen bonds than the 3. So we have helicase, and it's going to split the DNA. It's going to break those hydrogen bonds, wedge itself in there, and now it's going to create an opening. And we are going to create this little bubble, and we're going to hold the bubble open with these single strand binding proteins. So single strand binding proteins are these guys here and here. That's an SSBP. Single stranded binding proteins keep the bubble open, because otherwise DNA would want to coil back together and come back together and fix those hydrogen bonds. But we're not gonna do that with the single strand binding proteins. Those are gonna keep DNA open. And now we want to start to actually do the purpose of DNA replication and actually put down some DNA. So to do that, we are going to need a primer. So there's a primer, RNA primase. RNA primase is going to lay down RNA that is going to allow for the binding of DNA polymerase. So that DNA polymerase is going to bind onto the RNA primer. Where's our little RNA primer? Here he is. Here's an RNA primer. And it's going to bind on and lay down some RNA primase. And now we've got the layer down for DNA polymerase 3, if you really want to get specific. Then the DNA polymerase 3 lays down DNA. That's why we got RNA primase, lays down a little RNA strip of primer, RNA primase. DNA polymerase lays down new DNA. That's its job. So DNA polymerase 3 is going to lay down new DNA. Very important to note, when it lays down new DNA, it has to lay it down in the 5' to 3' direction. That's the only way this bad boy can lay down new DNA. So on this strand, it's called the continuous strand, the leading strand. It is going to lay down new DNA, and it doesn't have to stop. It can just chug along like a choo-choo train, just going 5' to 3', because over here is 3' end, and remember we said these things run anti-parallel. So on the leading strand, DNA polymerase just rocks it out, and it goes through the next step, which is elongation, and it creates DNA. So we're on to step 2, where we're actually running through and creating new DNA in this replication. So step 1 was initiation, when we had to get all the steps in order so we could actually start to lay down DNA for elongation. So DNA pol 3 lays down DNA. Leading strand, again, no problem. It can chug along. But the lagging strand, those guys run in the opposite direction. So what DNA polymerase does is that it's going to have to make it in strips. It's going to make it in these strips, and these strips are called Okazaki fragments. And so it comes along, it'll bind to a primer, and then it will lay it down 5, 2, 3, and then so it's laid down this one, and then it hit another primer, and then it goes and lays another primer down 5 to 3, and keeps going. So it has to lay it in chunks. Here's one fragment, here's another fragment, here's another fragment, because again, this stuff is running in the opposite direction, and so DNA pol 3 can only lay down in 5 to 3. Test question, only lay down 5 to 3. So leading strand's great, never has to stop, just keeps going. Initiation is where we're going to start this replication process. Starts at the origin, we go through the steps of initiation, and we start cranking out new DNA with DNA polymerase. On the lagging strand, we got to lay it down 5 to 3 still, but it lays down in chunks. Those chunks are called Okazaki fragments. And then we are going to have DNA pol 3 come along and remove the primase strips and fix those with more DNA. And ligase comes along, and ligase is going to, where's my ligase? There it is. Ligase is going to glue the little pieces together. So we have fragment, we gotta glue that strip together. That's ligase. So ligase is our glue that glues the DNA pieces together. It glues all those Okazaki fragments together. The last part of it is termination, and it doesn't go into a whole lot of specifics on termination. It does mention with prokaryotes that, remember, prokaryotes had circles. So when they start at their origin of replication, eventually you're going to end up with two DNA molecules that are like this at the origin, and then they wrap around, you got these two circles stuck together. So on termination, it mentions that topoisomerase 4 is going to come along and it will cut the DNA, and now the other loop can come free. That's topoisomerase 4 helps with termination on that aspect for prokaryotes. So again, know our 3 steps. Initiation is number 1. It's going to start at the origin. We're going to go through all of these enzymes. So topoisomerase unwinds it. Helicase wedges in there and splits the DNA. Single strand binding protein comes along and keeps the split open, that replication bubble open. RNA primase lays down the primers for DNA pol 3 to attach onto. DNA pol 3 will crank out brand new DNA. It cranks it out 5' to 3' every time. That's the only way it lays it down. We can move into elongation. That's where DNA pol is, that's its moment. And it's going to create a leading strand that rolls along just fine, no stopping. And there's a lagging strand that jumps and jumps and jumps between Okazaki fragments. And those fragments will be put together. We're going to have DNA pol that's going to come along, remove the primers, lay down DNA, and then ligase will glue the two little fragments together, and these two fragments together, and those two fragments together, until eventually you got a whole solid piece of DNA, both the new and the old on the bottom strand and the new and the old on the top strand. And then at the end, step number 3 is termination. So these are the 3 basic steps: initiation, elongation, termination. And then we've mentioned some of the enzymes here. This video goes into detail too. If you like YouTube videos, I know I like to have them to supplement stuff too. Here is a table of all of the different enzymes involved in DNA replication. Let's compare prokaryotes versus eukaryotes. There's a few things. Prokaryotes have one origin of initiation where they're going to start. And then eukaryotes have multiple origins of replication. So we can see all of these. Here's one, here's another one, here's another one. So we have lots of them. Eukaryotes versus prokaryotes, we have lots of origins. Prokaryotes just have the one. And again, the origin is where replication starts. Another difference between the two is that the rate of replication. Prokaryotes can replicate super fast. Eukaryotes not as fast. I believe it said in the textbook, prokaryotes replicate their entire cell in like 42 minutes. So their rate of replication is much, much faster than ours. Ours is 10 times slower. Eukaryotes are 10 times slower than prokaryotes. So that's another difference between prokaryotes and eukaryotes. One more that they mention, prokaryotes have circular DNA, right? Eukaryotes don't. We've got linear chromosomes. With our linear chromosomes at the ends of them, we have these non-coding segments called telomeres. Telomeres, because at the ends, every time we copy, eventually a chunk falls off and a chunk falls off and a chunk falls off. Because we have this linear structure, we are going to, there's no place for that DNA primer piece to be copied on, and we lose a little piece every time replication happens. So we have these non-coding segments at the end. It could be 100 of them, it could be 1,000 of these non-coding segments, but we got a lot of them so that it can copy itself and not risk losing genes, at least for 100 copies. So you're gonna see these telomeres at the end. There's something that can put them back on, and that is what Elizabeth Blackburn discovered, and that is telomerase, which is an enzyme that puts these repeating segments back on. So telomerase is added on this segment here and adds it back on this segment here. So basically it can just keep going and going and going and going, and it won't run out. That's kind of how it would have uncontrolled cell growth if this kept happening all the time. Our stem cells for eukaryotes, our stem cells will do this. Our normal adult cells that are not stem cells, that are not germ cells, our normal regular somatic cells, they won't. So they will have a limited lifespan on these things. But germ cells will have telomerase that could tack on those telomeres, and the cell can continue to copy itself without fear of running out of DNA and eventually losing the genes, and that cell dies. So this is telomerase. It was discovered by Elizabeth Blackburn. And so she got the credit for that in 2009. Looking at all of the table that compares the two, again, we've got one origin at prokaryotes. Eukaryotes have lots. Maybe 100 nucleotides per second, 10 times that for prokaryotes, 10,000. I'm not going to ask you about all the different DNA polymerases. No. Okay. Yeah, that's my big takeaways. I want you to know that there is just there's one origin there, and we've got lots, that they are much faster than we are at copying the DNA. If you want some real specific info on, real detailed info on comparing prokaryotes to eukaryotes and the differences in regards to replication, watch that video because he goes into lots of details, way more than I do. Rolling circle replication. So this is used, this is used by plasmids, because we said plasmids are the extra chromosomal DNA that bacteria can swap back and forth. And usually it's extra cool stuff like antibiotic or toxic genes to release, what was it, colonizing factor, SL and LT or something like that. Those toxins that can be produced. Maybe that's found in a plasmid. And if I share this plasmid with my fellow neighbor bacteria and they take this plasmid, then they also have these genes. So plasmids will undergo rolling circle. Some viruses do rolling circle too. And this is the way it's going to happen. So we said that there was one origin, right, on replication. So we are going to have origin. We got our double-stranded origin is right here, and it's going to start with a little enzyme that comes along and cuts it. So the enzyme nicks it at the origin, and now we are going to bind on to DNA pol 3 because DNA pol lays down new DNA. So DNA polymerase 3 is going to bind on that origin, and it's going to lay down new DNA. And I can cover this up because it's already red. So DNA pol 3 is laying down new DNA, and every time it wedges through here and lays down more DNA, it shoves out the old strand. And so it's literally like a rolling circle, and it's peeling away the older DNA strand as it lays down new stuff. So new stuff goes around all the way. It's going to go all the way around in this circle. And then eventually it's going to finish out that circle. It's in a rolling circle. And here is our completed double-stranded DNA synthesis. On that first strand, we have this other lone strand out here. So right now it's a single-stranded DNA molecule. If that single-stranded DNA molecule is lucky, it's going to meet up with another enzyme, and that will allow it to bond, and DNA pol will lay down more DNA on that guy. So this is rolling circle. It starts with a little nick, and then DNA pol wedges in and creates more DNA that's going to peel off that outer layer, and we're going to end up with two plasmids at the end. One of them will be double-stranded from the get-go, and the other one is single-stranded, and it needs to have another molecule, another primer and polymerase to tack on to land to make this circle complete. So that's the first two sections of chapter 11. We will talk about RNA transcription and what we do with that when we transcribe it from DNA to RNA. We'll cover that in the next section.